Hello and welcome. I'm Karen Panetta and I am the IEEE Awards Board Chair. Today, joining us, we are here today to celebrate the 2022 IEEE Francis E. Allen Medal, sponsored by IBM, to Eugene Wimbley Myers Jr. and Webb Miller. Joining us here today is Eugene Myers Jr., and I want to first congratulate you on this great accomplishment.
Thank you. I'm very pleased to be winning the award, especially with Webb Miller, who's been a longtime friend and a wonderful colleague.
Okay, and joining us here today are some of our IBM representatives. So I would ask them to introduce themselves, starting with AJ.
Thank you, Karen. Hi, Jane. I'm Ajay Royuru, Chief Science Officer for the Healthcare Life Science Research in IBM. I see Jeff Welser has joined. Maybe Jeff, you go next.
Sure. Hi. Welcome. Thanks very much. Congratulations, Gene. I'm Jeff Welser. I'm the COO of IBM Research. I'm also in charge of our exploratory science and university partnerships. Lakshmi, I believe you're on mute.
Sorry. Congratulations, Gene. You know me, but I am Lakshmi Parida, and I'm leading the computational genomics effort and also the exploratory life sciences in IBM.
Hi, this is Tanvir Saidar Mahmood. I'm an IBM fellow here on the West Coast, and congratulations, Alan, for winning the Francis Allen Award, Eugene. I'm a long-term follower of the bioinformatics area, although I don't work in this space, but I'm trying to see some exciting connections between that and imaging work. So I'd love to ask you some questions during this session. Thank you.
And hi, Gene. I'm Jeff Rogers. I lead our digital health efforts at IBM. Congratulations to you and Webb Miller for the great accomplishment.
Thank you. And I'm especially delighted that the award is in Fran Allen's name. You know, I knew of Fran's work when I was a PhD student. You know, she was already a star for me. So, you know, for me to win an award in her name is special. And thank you also to IBM for you guys choosing us. Appreciate it.
So let's start out for our audience, Eugene, to talk about what, you know, you've made some seminal contributions, and genome sequencing is something that years ago when we even talked about it, people would say we were crazy, it couldn't be done. And you had the insight not just to start working on these problems, but to share it and make it open source. Can you just talk about what inspired you to even go into this work and how you got started?
It was actually a coincidence. I was looking for something interesting to think about. I was a computer scientist back in the day. This was back in the early 80s. I was, you know, I just done some work on diff, on comparing strings. And so I was quite interested in things about strings. And I met a molecular biologist from across the mall at University of Arizona. And he said, well, there's this interesting problem, you know, it's assembly. And I just thought it was a great problem. I mean, I was kind of focusing on how to put the pieces, and it's just, you know, it's serendipity basically. But it's turned out to be a fascinating problem. You know, people are still working on it, actually, algorithmically. It's not over yet. But I'm happy to have played a role.
Okay. AJ, you had a question for?
So Jane, congratulations. This is, of course, very fitting recognition. Like you said, the connection to Fran Allen and, your inspiration from her work. If she were here, she would be equally celebrating the fact that you have built upon her work to actually do this seminal work. And, you've really inspired actually the whole field in thinking about how algorithms get applied to problems of sequences. And, over the decades of your contribution, it's just amazing that, it actually goes into so many application areas. But things that stand out really are, in my mind, looking at it from applications, alignment and assembly were kind of problems that people didn't even think would be solvable. And, you and Webb have come at it with such force and inspired a whole field of work. So I think Alan would say that as well. Fran Allen, if she was here, she would say that. I have a question for you.
Sure.
So say what your feelings were back then in tackling something that hard. How do you even consider that to be a solvable problem before you solve it?
Well, I mean, originally I didn't actually see it. I mean, there were a couple of contributions. I mean, one was the search engine BLAST, which I think is one of the things that Webb and I both contributed to. And then there was the work on whole genome assembly. So, I mean, I'm very happy that basically we were able to prove that computer science was worth something. I mean, the contribution for BLAST was that we basically, as computer scientists, saw how to basically lever the information in an algorithmic way that basically vastly accelerated the search for these sequences. That was the problem. They were at the time in the 1990, they had a search engine on the growing nascent databases, but it wasn't fast enough. And it was an approximate match, but looked like a very hard problem. And, I won't delve into it, but basically Webb and I had the machinery and the insights from our work thinking about comparing strings to basically figure out how to build a very fast heuristic. And, what was really powerful about that was that we sped it up a couple of orders of magnitude.
Yeah.
Had made kind of regular search at the NCBI and the national database as possible. So that was kind of the first big accomplishment. In fact, that paper is like one of the
most cited, yes.
Cited papers
Yes.
in the top 10. I mean, the amazing thing is I was oblivious. I was a young kid. I was an assistant professor. We did this piece of work. We wrote this paper. And I didn't have any idea the impact it was having, actually. I went on to the next thing. I was working with my PhD student, John Kechiciogu, on assembly. And it wasn't until there was a U.S. News report about rankings of computer science. They compute all these statistics. And Arizona was suddenly like number 2 or 3 in citations per faculty member. We were all scratching our head going, how did we get to be number 3? And it was this one paper. Yeah. And that was the point. So it was like 3 years later, I suddenly realized, wait a minute, people are citing this thing like crazy, and it of course it makes perfect sense because everybody was doing a search. Yeah. Every paper involved.
Exactly. Yeah.
So they cited the paper. So, we were getting, thousands of citations. So that was the first one. I'm sorry, is the answer too long? Should I shorten it?
No, we want to hear this, but we want to get our papers cited and get our institutions elevated to the top notch.
Yeah, a lot of success is just good luck and being prepared when the opportunity comes. I mean, I think really when I look back at my career, the assembly thing, I mean, what was going on with assemblies kind of in the mid 90s, kind of everybody, there was this problem. It was kind of a jigsaw puzzle. You wouldn't actually get the DNA sequence kind of from left to right. You would get like pieces of it. You would get pictures, windows of it. And you had to figure out how to put the pieces together. So it was like kind of the analogy would be to a jigsaw puzzle. And the feeling at the time was that that algorithmically, the biggest kind of jigsaw puzzle you could solve was about 1,000-piece puzzle, which to me is kind of interesting with the analogy of human puzzles, right? It was like literally 1,000 pieces, which I thought was kind of funny. It was, but it was about 100,000. And so, to do the human genome, which was 3 billion, right? Everybody was thinking that we were going to have to figure out how to, break the human genome down into pieces that were solvable, 100,000, and kind of figure out how to tile those together and then do it. A very complicated thing. I mean, secure, but complicated because it's a 2-stage process. And basically, what the kind of the revolutionary thing was is basically I said, along with Jim Weber, who was at the Marshfield Clinic, who proposed this with me, was like, no, let's just shotgun the whole thing, it's a 50 million piece puzzle. With the one caveat that we would have information about how the reads were linked. So we called it paired end shotgun, because we had the ends of sequences, and so we had these pieces of data where we knew kind of the distance between them. So imagine having pairs of reads, of data, of strings with a known distance. And by building a library of distances that spanned the typical repetitive elements in the genome, because you see the problem was what made the genome difficult is that there were things called signs and line elements, which were sequences that would occur 10,000 times or, you know, a million times in the human genome. And so these strings all looked the same at the resolution of the lens of the individual reads. But with the links, it was suddenly possible. I mean, I won't belabor it too much, but we proposed this and everybody thought it couldn't be done. And again, I won't belabor it. I mean, basically the trick was to figure out which parts of the genome were repetitive and which ones weren't by looking at kind of certain statistical aspects of the coverage of components of that. But if you could kind of separate who was repetitive and who wasn't, which is kind of the key trick that I solved at Celera, then the whole thing comes out, okay? And the cool thing is analysis is the way everybody does it. So that was kind of an, you know, like that one I say was luck, you know, it's like I proposed it with Jim and, you know, to do the human genome and we were completely shut down. I mean, completely shut down.
Right.
They didn't even invite me to the meeting at, you know, the island where they set the standards. They only invited Jim and they, you know, they tore him to shreds. He's a geneticist, he's not a statistician. And then, you know, I was kind of cruising around Europe trying to get people to do a pilot. I thought, well, okay, we won't do a human genome, but how about we do a fly this way, or at least try it, you know, because it wasn't 100% clear that it could be done. I mean, I thought intellectually that, you know, and I'd done simulations and people were debating me about the simulations, but then it was like they decided to form Celera, right? Which was like, you know, they put 300 million into Celera. And it was just, to me, it was like they had basically decided to fund my idea, you know. Like, so I was basically one of the first, you know, 10 people through the door.
Yeah.
I took the chance, and then the rest is, yeah, you know how it goes.
Yeah, so that's fascinating, Gene. In fact, when you're saying, when I was at NYU, at that time I was in graduate school, and I think the understanding there was that it's not possible to do it this way. So it's actually very impressive that you just sort of took it to completion by following your method. But Gene, so when it comes to string algorithms, assembly, and so on, I know you covered that space, and I have attended talks of yours recently where you're still, like you said, the problems are still there, and you're still talking about more efficient assembly and so on. But I also know you're doing, I think I know you're also doing a similar thing about imaging embryos, getting the images from all angles, and trying to map the embryos of living things. So do you want to say a little bit about that journey?
I got very interested in that after the genome. I mean, so after we did the human genome, the questions, you know, it was like we had the sequence, but we didn't know what it did. And what occurred to me is that the most natural way to understand what the human genome did, or a genome did, was to basically look at the developmental trajectory. I mean, the genomes encode about 50% of the coding potential of the human genome is there to make you, okay? And the other 50% kind of maintains you. But a lot of it's there. So if we want to understand a lot of what was going on, we just needed to watch things being made. Now, we obviously couldn't watch humans being made, but we could watch flies and other things being made. And one of the big things about having the genome is that once we had the sequence, then we could go in and we could light up any protein or kind of genetic element that we wanted, right? Without the sequence, you don't know how to target your fluorescent molecules. But once you have the genome, you basically have direct access to all of it. So suddenly, I mean, one of the biggest outcomes of having the genome was that we immediately were able to basically immediately able to instrument it. and explore it and, knock things out and knock things up and light things up. And, what I saw was is that a lot of the experiments around that were basically visual. You put them in a microscope, you light something up or you knock something out, and you look at it, and you watch it, you see what happens, right? So you do deltas, basically. You're doing kind of, you're making your, hidden parts of this black box and seeing what happens. And what occurred to me is that nobody was working on computer vision. Bioinformatics was primarily the domain of people that thought about discrete things, sequences, graphs, discrete mathematics. And suddenly here's this thing that really requires continuous mathematics. And it really is the case that, the engineering and the mathematics behind computer vision is continuous mathematics. It's quite different. And so despite that, though, I thought that it was a very important component of computational bioinformatics. It's a computational problem.
Wow.
We needed to basically be able to watch these cells, track them, and there were very few people working on that area. So I basically thought, and the other thing is in the meantime, so that was the pull. The push was that they basically stopped doing Sanger reactions and they went to doing short reaction resequencing. And so suddenly, to me, the assembly problem was completely uninteresting because with short reads, you can't possibly do what we did at Solera. So it was kind of like we went into this. I'm not saying that it wasn't incredibly important, but the emphasis became on how to resequence people using short reads. So you already have a human genome and you're just trying to figure out. So you wanted to understand human variation. Very important medically, but for a guy who's interested in kind of, combinatorial problems, the data doesn't go together well, and I was like, well, okay, that's not going in a direction where I can do anything. So I said, so that was the push, okay, and the pull was, yeah, this is cool. Look at this. It's been, so I started doing that in about 2005, Lakshmi, and honestly, it's been much harder, okay, than the sequence stuff. It's a much harder domain. I think my work has had some impact there, but these computer vision problems are very, very difficult because they are intrinsically things where you want an AI. I'll leave it at that. To mimic what the human eye system does, and software is incredibly hard to do. But we've had some luck with methods that are combinations of combinatorics and together. I won't belabor it. Although honestly, I'll finish now. I just retired actually as a Max Planck director on April 1, and for the remainder of my career, I've decided to go back to focusing on sequencing. And if you want, you can ask me why I'm back in the sequencing game. And so I've done a few more pieces on the computer vision, and I think set that in a good direction. And now I'm gonna finish my career answering the last parts of the assembly problem that I feel are still unsolved. That's the plan anyway.
Fascinating. I think the connection you made between vision and biology, this is actually, the field is going that way now. If you look at many of the top conferences, I just ran ISB conference recently, and then one of the topics that we were seeking submissions and was showing the interplay between looking at the holistically for a particular disease, the evolution at every level of observation, starting from the genomic to proteomic to cellular to pathology to radiology, and going across scales is the thing nowadays, and fusing across multiple modalities is a field of research both in our emerging areas, but also in core AI as well. So this is, I think, very germane to, and maybe new advances will come with people like you continue to focus in this particular area. But one of the questions I did have, and I see the sequence alignment and the complexity, the inherent complexity of BLAST and using underlying dynamic programming algorithms and so on. with the evolution of the current generation of transformer models, and you now see them used in NLP and applied to bioinformatics and all, what is your opinion on, is there any fundamental change in the complexity do you see emerging with the use of neuronal architectures, and do you expect to see new discoveries with the transformer-like instantiations of algorithms and their variants subsequent to BLAST, like approaches of the past?
So sorry, your sound broke up a little bit, but you were asking about are there transformative aspects to the
With the use of neuronal architectures like the transformers and their use in sequence alignment and addressing problems in this space, do you expect to see some new discoveries happen, or you see that the fundamental problems are still the same?
Yeah, I mean, things are mostly incremental in my experience. I mean, there's every once in a while there's a jump, and those are always surprises. So I can't tell you where the next jump is going to be because I don't have a magic crystal ball. So forgive me. But you know, I use deep nets and AI techniques. They're very valuable. Basically, a deep net can learn any continuous function if given enough training data in a way that nothing else you can algorithmically. And that's extremely useful. And in fact, that's one of the reasons why maybe it's a little bit too technical a point, but that's why they're so useful in computer vision, is because the function, they've got a built-in regularizer in the sense that they only compute continuous functions. Okay, so they're already regularized in a sense. So they have a prior. But my own experience is that nets are very sensitive to what they're trained on. And so they can be incredibly powerful if you have a very controlled domain. Like AlphaFold, for example, was tremendous, but it's a very controlled domain. I mean, there's only about, 30,000 different, protein sequences. I don't know, maybe 500 basic shapes. And, you're talking about 20 amino acids, and you have a very controlled data set with very good information, ground truth information. And so they're, and the domain's the same. You give it a new protein, it's not like you've got new characteristics. For example, we used deep nets to do deconvolution on images. Beautiful. If you train the net on a given microscope and on a given experimental rig, I'm looking at a Drosophila embryo, and you train that thing and then you run it, beautiful. Okay, you go to a different microscope and you use the same net, it doesn't work, okay? I mean, it works, but not great, okay? Or you do a different set of conditions, you do a C. elegans embryo, slightly different reagents, different diffractive code, doesn't work as well. So that's what I mean. It's very sensitive to training. In fact, when we put out this deconvolution algorithm, we said, very careful, big caveat, train it on the rig that you're going to be using it on. Okay. So I think nets are really tremendous in that they can do things. And, I guess the question is, are we really going to get to AI? Right now, I mean, deep nets are an incredible way of learning, like I said, a continuous function. They learn how to classify. Are they doing what we do? Not yet. But it'll be interesting to see. I mean, to me, the question is it just a question of scale? Are we so special because we have 10 billion neurons instead of the billion that a chimpanzee has? Or are we special because something happened on the way it was wired? Don't know. We'll find out. But you'll find out. I may not live that long, but I'm sure you'll get the answer.
Thank you. I'll let others ask the question.
So if I might, I think one thing I would highlight that I'm hearing you talk about is the importance of picking a problem. And as you said, right, you don't know if it's going to be the right problem, right? A lot of it seems incremental. and then every once in a while you can make a jump in there. But given your perspective now, because you've had this opportunity to make big contributions, where do you see interesting problems moving forward? For the people who are listening, what are some areas where you see opportunities? It doesn't have to be in what you do exactly, or it can be. I think you highlighted one just now about AI. What are some that stand out to you? Maybe what do you see?
Well, actually, I want to actually emphasize something else you said, which I think is very important from a mentorship point of perspective, because I tell a lot of my graduate students, as I said, at some point, everybody can solve a problem that's in a textbook. Okay, so you get to the I can solve the most difficult problem at the textbook level. Okay, but that doesn't make for a successful career as an academician or as a scientist. What does is your ability to know what to work on. Mm-hmm. And that is actually more important than anything else. And that's what I learned over the years, is that it's problem selection and quote unquote aesthetics that are very, very important kind of move, and you don't get it right, you're not always on the way, but so another thing I always say is never work on one problem. Mm-hmm. You should have at least a half dozen problems that you're working on at any time, because you never know which horse is going to come in. Okay. So, but anyway, so the original question was, where do I think it's going to be interesting? Well, oh dear, I'm going to run out of power. They kind of caught me. Hold on a second. You're
going to tell us the secret of the universe.
Okay, there we go. Okay.
Power at that moment.
Now I can regale you for the next 2 hours. Okay. Everybody wants to stay on, right?
Right.
No, I'm kidding. Where do I think it's going to be interesting? Well, I think in terms of, you want to know in terms of molecular biology and the interface with computer science, right?
Sure.
I can speak to that. I think there are a couple of interesting areas. One is our recent ability to basically generate what are called organoids, or synthetic developing embryos, because it's basically tissue engineering. So a lot of the work that we've been doing in molecular biology has basically been doing these deltas and seeing what happens. But suddenly we're actually able to create synthetic systems, these organized systems. We're basically able to take a cell, take it back to the stem cell state so that it tries to make a brain, or it tries to make a liver, or tries to make a heart. And studying these I think is going to be a huge new dimension in molecular biology in general. And I think that, one of the real challenges is that we can't actually, informatically, it's hard to extract quantitative information from these things as we watch them under a microscope. Most of the observations that have been made so far have been quantitative, because it's very hard to model these things. So I think that, I do think that there's going to be substantial advances in light imagery. And I think that's going to open up new problems in computer vision. It's speculative, but I believe that looking at what optical physicists have been doing with adaptive optics and things, that we're going to actually be able to get a much clearer picture of these things. And I think that's going to open up a lot of interesting room for analytic tools. The other thing that's happening is, nobody asked me a question about why I re-entered sequencing, but basically, we now have a third generation technology of sequencers, which produce actually not very short pieces, but very big pieces, and very accurately, actually, to the point now where we can basically say, look, I'd like to know what the genome of that, aardvark over there is, okay? And we can take some blood and we can extract the DNA and we can run the machines and run the assembly algorithms that we have today and get almost a perfect reconstruction of that genome, much better than the ones we were getting back in 2000 when we said we sequenced the human genome, right? These things are nearly like telomere. So we went from sequencing the human genome and the fly and a few other things at a high fidelity. Then we went into this period where we resequenced human beings like crazy and kind of produced crappy assemblies of things that people were interested in, but enough to still see the genes and things. I mean, to do interesting science. You know, you got a hammer, you use it. But now we're back to the point now where we can produce genomes that just, you know, that make the Celera result look kind of hokey. Okay, I mean, it's so good. Okay, and so what's happening is there are consortia developing to basically go out there and sequence the natural world. There's the Earth Biogenome Project, which is kind of an umbrella organization, which is trying to organize all of the people that are trying to go out there and sequencing things. Mm-hmm. There's groups that are going out to sequence bats. There are groups that are going out to sequence all the vertebrates. And so this is going to be fascinating biologically. I mean, I could spend a little bit of time, but to get to the reason why would it be interesting to an informatician is that, okay, you know, we've kind of been working for the last 20 years on how do we align, you know, 100,000 human beings, where our sequences are almost all alike, by the way. They're 0.1% of that easy problem. Okay, how now do I take 100,000 species? There are 1.5 million extant species of life that are cataloged on the planet, by the way. If I had those, how would I compare them? Okay, so I mean, it makes the early comparison problem seem really easy, right? And scalability, and how am I going to do that fast enough? And I mean, geez, that's a big challenge. And I think, and I do think that that's where genomics is going. I mean, there's still going to be a huge amount of work on people, on medicine, because, you know, we all want to live longer and better. But scientifically, we're going to go out and sequence everything that's out there that we can, you know, both for ecological and, you know, preservation reasons, and also because it's actually, if you want to know what a genome does, actually, it's better to do deltas on things that are interesting than they are on human beings. Okay, so I'll do one more little bit. Okay, so human beings are, you know, we're the result of a very narrow bottleneck event. I mean, there were maybe, you know, 10,000, 80,000 of us that kind of came through filtered as modern man. Okay, and we've spread since then, but we're almost all alike, okay, genetically, all right? It just happens that we have brains that sense things like, you know, skin color, or, you know, things like that. But that, frankly, in terms of genetic content, those things that we describe as ethnicity are nothing, okay? Which kind of says, why is it a big deal? Okay, well, the reason it's a big deal is because we're tribal, and our brain has learned very much to recognize the tribe, and that's why. But if you think about it genetically, okay, it's a nothing burger, okay, but that's exactly the point. I mean, if you want to study heart disease or longevity, for example, how long do people live? Well, you know, absent disease, absent somebody getting some chronic disease, you know, people live the same length of time, plus or minus 10%. Okay, so we're talking about, you know, if you look at all the genetic data, you're seeing a 10%. I can find a pair of bats that are just 5 million years diverged, which is nothing in terms of evolutionary time, where one of the bats lives 50 times longer than the other one, okay? So I want to know, and in fact, I'm working on it right now. We're sequencing these things. I want to know what's different in their genomes, okay? And I'm much more likely to find something that tells me why the one lives 50x longer than the other, because the phenotypic difference is huge, right? So a lot of what we're trying to do is we're trying to understand the genotype to phenotype correlation. We're trying to understand how the genetic sequence determines the physical presentation and attributes of the creature involved, okay? And the best way, you know, you guys are all mathematicians, you understand that the best way to do that is when the difference is as big as possible. Yeah, it's that simple. So I think that these projects to go out there and sequence the natural world, they're going to, I would predict that they're going to revolutionize medicine for human beings. Okay?
And so we know, so Gene, we know that you're really not retiring from this discussion. So we look forward to seeing all these great projects. I want to know about the bats. I'm looking forward to seeing the discovery on that one. And at this time, I do want to thank everybody for a wonderful conversation. And Jean, we want to congratulate you again on being the recipient of the 2022 IEEE Francis E. Allen Medal, sponsored by IBM.
Thank you, guys. Yeah, thank
IBM
you.
representatives. And I look forward to seeing all of you out in California at our IEEE Honors and VIX Summit. Thank you. Bye, everybody.
Thanks for listening.